Hello, everybody. Welcome back to the second session of Agrigenomics track on day one of the Asia Pacific Japan Genetic Solutions Tour. For those of you who joined in our first session, we are talking about the axiom microarrays and how they are being used for various genetic applications. My name is Srinivas Chadram, and I am the Global Product Manager for Agrigenomics. And in continuing our focus today on the Applied Biosystems Axiom Microarray platform for agrigenomic applications, we have our next speaker, Dr. Rommel Herrera from Philippines. He is a scientist at the Philippine Carabao Center at the University of Philippines in Laguna. And today, Dr. Herrera will speak to us about how his group has used genomic information to tailor a breeding program for the Carabao, the native buffalo of Philippines, and how the Applied Biosystems Axiom platform was customized for his breeding analysis for improved milk production. So over to Dr. Herrera.
Good morning. I am Rommel from the Philippine Carabao Center, and I will be presenting my research on the use of genomic information in the dairy buffalo breeding program. Basically, this is a collaboration between PCC and the University of New England, led by Professor Julius Vanderwerf. This is mainly funded by the Philippine Council for Agriculture, Aquatic, and Natural Resources Research and Development of the Department of Science and Technology. The government, through the Philippine Carabao Center, has undertaken a genetic improvement program for dairy buffaloes. Selection of dairy buffalo bulls is based on the performance of daughters, basically for the testing. The Philippine Carabao Center has 12 institutional herds spread throughout the archipelago. This houses around 1,200 females, around 400 are considered unique dams. Also, we have 2 bull barns each with a semen processing unit. This houses around 63 bulls, 3 proven bulls, and around 60 mating bulls. So in the Philippines, we have 2 types of water buffalo. The Philippine swamp, we call it the Carabao, and we mainly use for draft work. And then we have the river type, the Riverine type of buffalo, which mainly is used for milk. We imported first the American Mura in 1994, and then the Bulgarian Mura around 1995 to 1998, and then the Brazilian Mura in 2010, lastly the Italian Mediterranean in 2013. This is mostly what we keep in our institutional herds. Most of our semen donor bulls now are of the Bulgarian Mura type, and we use this to inseminate mainly the swamp buffalo. So the process have better growth and better milk production. So that means it translates to better income to the farmers. Our breeding structure is shown. So as mentioned, our institutional herds are spread throughout the Philippines. Can be divided into nucleus herds and multiplier herds. Also shown are the 2 bull barns with the semen processing units. So the top tier contains the nucleus herds. These are mainly the Riverine buffaloes. The second tier contains the buffaloes, again Riverine, which are in the multiplier farms. The lower tier contains Riverine, crossbred, and swamp buffaloes in the dairy cooperatives and individual farmers. The flow of frozen semen goes from the nucleus herds down to the multiplier and the dairy cooperatives and farmers. In terms of the flow of heifers and bull genetics, used for natural service comes both from the nucleus herd and the multiplier herds, and it goes down to the multiplier herds and the multiplier herds and the data producers and the farmers. However, present constraints include the following. As mentioned, the carrying capacity of institutional herds is only for 1,200 recorded cows. And we have limited space for progeny testing. We only progeny test around 8 bulls per year. Moreover, accuracies of progeny test bulls are low due to the small number of daughters with lactation records. And progeny testing is slow. It takes approximately 7-8 years to identify the best bulls. And with this very long process, it's very expensive. Okay, I now present you the schedule of progeny testing activities. So it takes around 100 months to identify the best bulls. Please take note that the water buffalo has a longer gestation period, around 10 and a half months. And then we could only get semen from young bulls at around 3 years of age, 2.6 to 3.3 years of age. So it's longer compared to, takes time compared to dairy cattle. However, with the above constraints, so typical for a developing country, the phenotypic trend for our population is going up. So it, on average, it increases 19.5 kilos per year. In terms of genetic trend, for milk yield, fat yield, and protein yield, it's increasing. So for milk yield, it increases 8.9 kilograms per year. For fat, it's 0.89 kilograms per year, and for protein, it's 0.37 kilograms per year. Not bad. Now, in 2013, the buffalo snip chip came out. It came from Affymetrix. It's the first commercially available and so far the only available snip chip for buffalo. Contains 123,040 probes representing 90,000 SNPs. The total length covered by the SNPs, or the genome coverage, is around 2.658 billion base pairs with an average interprobe spacing of 21.6 kb. Now we use this in our population. So we tried this on the 4 Riverine breeds and from the Philippine swamp. So, okay, we have 3 columns representing the 3 informative SNPs: the polymorphic high-resolution SNPs, the no minor homozygous SNPs, and the monomorphic high-resolution SNPs. As can be observed, in terms of the Riverine breeds, they have a more number of polymorphic SNPs, on average around 75%, compared to the monomorphic SNPs, on average 7.6. However, in the case of the Philippine swamp, most of the SNPs are of the monomorphic type, 55,000 or 61.2%. They have a lower number of polymorphic SNPs, only 13,929 or 15.5%. So we used around only 261 buffaloes to determine the polymorphic SNPs from our local buffalo populations. So we generated a PCA plot of animals belonging to the Riverine buffalo populations and from the one swamp population. As can be seen, there's a clear separation between the Riverine populations, Riverine type buffaloes, and the swamp populations. So EC1 is 96.9%, accounts for 96.9% of the variants. So this is swamp, buffalo. The blue one is the Bulgarian Mura. The red one is the Brazilian. And the yellow one is the American. So they group together. This separate group is the Italian Mediterranean. So we did another PCA plot showing only the 4 riverine breeds. So clearly there's a group formed by this the Brazilian, the Bulgarian, and the American. And the Italian is separate group. This can be explained by the histories of the breeds. The Bulgarian Mura is 75% Indian Mura, 25% Mediterranean. The Brazilian animals that we imported are mainly Indian Mura and their crosses. And the buffalo from America came from Florida. Again, we checked the history. It originated from India. So basically, this group of animals, these three breeds are Mura-based, while the Italian Mediterranean is a separate group. It's mainly Mediterranean breed. So we also got the LD for the different breeds. So we got the minimum distance between markers for effective coverage for genomic selection. So Bulgarian and the Brazilian around 40 kb, and the American around 200 kb. So the average intermarker distance is 44 for the Bulgarian, 43 for the Brazilian, and for the American is 54.8. So basically, I'm saying is the markers are dense enough. There is a sufficient LD to do genomic selection using the buffalo snip chip. Okay, so from that information, a reference population for genomic selection contains, again, this group of animals: the Bulgarian Mura, the Brazilian Mura, the American Mura, and their crosses. Bulgarian cross to Brazilian, Bulgarian cross to American, because mostly we have Bulgarian semen donor bulls. And the reference population, since we have very few proven bulls, we will use all females. And these females will have either 1, 2, or 3 lactation records per animal. Now, for genomic selection, with the reference population, all females, but we will do single-step G-block. We will include the pedigree, the phenotypes, or the performance data, in this case, milk yield, and the genotypes for the reference population. So we produce a prediction equation, and then the selection candidates are males with only the genotypes, because I will use these bulls, semen donor bulls, so I could disseminate the genetics faster to the rest of the population. So selection candidates. So in the end, I will have selected young bulls with genomic breeding values. Hopefully, there's increased genetic gain because the generation interval will be shortened from around 7 to 8 to 3.5 years old. And then, since there is no more waiting bulls, or continuous testing will be stopped, there's will be decreased long-term costs. So, so again, female reference buffalo population. I used 1,930 animals, mostly Bulgarian Muras, with 3,731 records. Only 859 were genotyped, with 2,642 animals in the pedigree. I used the buffalo snip chip, So this is basically single-step G-block. So male selection candidates, we use around only 12 DNA tested using the Snip chip, and we will have selected candidates with genomic breeding values. And there's increased genetic gain and decreased long-term cost. So why did we use single-step G-block versus the conventional block model? So in single-step G-block, wherein he used again the pedigree information, performance data, and the genomic information, there's the correction of some relationships of animals in the pedigree, because genomic information is more accurate than the family information. And then this links different families, again, because we will now use genomic information between animals. And then this additional SNP marker information provided by the genotype animals extends all to the genomic relationships with all the pedigree, thus increasing the accuracy of prediction. We did a threefold validation wherein the phenotypes considered were animals with 1 lactation or 2 lactations or 3 lactations in the test population. The accuracy we got for the conventional is around 0.17, but for single step, it increased to 0.30. So there's an increase in accuracy of 0.13 or around 76%. The reason for this is the above reasons: correction of the relationships of animals in the pedigree, and then linking the different families, plus the extension, the additional SNP information to all the genomic relationships. So this slide presents the scatter plot of breeding values from the single-step G-block compared to the breeding values derived from the conventional block animal model. So the x-axis contains the EVVs for P-block. The y-axis contains the breeding values per single step. So the correlation is 0.84%. So basically, I'm mainly interested in this animals here. These are all females. I'm only interested here. So there is the ranking among the top ranked animals. So 62% are both found in single step block, single step G block, and the pedigree block. Another representation is like this. So if I would only get the top 100 animals from T block, top ranking cows in T block, and the top ranking cows in the single step, 62 out of the 100 females overlap. So 62 of the females are both present in T block and the single step. So basically, there's a ranking. There's a ranking again because I used genomic information together with the pedigree information. So we did an analysis on the genetic gain and the savings, comparing the progeny testing versus the genomic selection. The genomic selection, around 22 to 33% increase in genetic gain occurs compared to progeny testing. It depends on the number of pools in the institutional herds, from 3 to 6. So that's why this range is present. But still, there's an increase in genetic gain if you use genomic selection. Now, in terms of savings, due to the reduction of number of bulls maintained from 63 to 40 animals, so basically I don't need the 23 bulls, there is a savings of around 121 million pesos, or 28% less in terms of net present value savings compared to PT. So we considered a 6,000 semen doses per bull, 5% discount rate, a 15-year time horizon. So if there's a ranking of females in the reference population, there is also a ranking of selection candidates. So these are the bulls we plan to use in our institutional herds. As you can see, these top 3 bulls are are the best ones for the T block, the EBB milk. But if we do the single step T block, the top 3 now would be this bull, and this bull. So there's a ranking, but more or less same bulls are identified. More or less same bulls are identified to be on the top. So these are pictures of our genomically tested bulls. This is Lucas, Donald. So these are both Bulgarian Muras because of this number. First number will indicate the breed. These 2 numbers will indicate the year born. So these are both 2015 born bulls. Here's Edu and Dexter. So again, they're both Bulgarian Muras. This is born 2014. This is born 2015. So we have 2 more bulls included, Warren and Jano. So they're both 2017 born bulls. But these 4 bulls are already are included in the mating plan for 2020. The last 2 bulls, both born in 2017, will be included later this year in the mating plan for the institutional herds. So future plans, so we need really to continue to increase the reference population in terms of the number of animals with performance data and the number of animals genotype. With limited budget, we could only genotype around 2 plates a year, so around 190 animals to be genotyped per year. Then also a need to develop a genomic prediction model for other traits, since we also collect phenotypes for this one, for fat yield, protein yield, and somatic cell count, and then there are some fertility traits. The Snip chip is a bit expensive, so it's around $200 genotyping cost, including shipping cost to the United States. So there's a need to develop a low density chip designed for the local population. So lastly, I would like to thank University of England for assistance, the guidance, the support. My Professor Julius Vandenberg, Dr. Cedric Mundro, Dr. Leonie Jumisting, Dr. Nassim Munir, the Philippine Carabao Center, Dr. Esther Flores, the staff of the Genomics Lab, all the coordinators, the herd managers, and directors of all the centers. I would like to thank again the USTP card for funding this genotyping project. And lastly, to CIRCA for funding my PhD scholarship. Thank you very much for your attention.
Thank you, Dr. Herrera. That was a very interesting presentation. Thoroughly loved it. Glad to see some work on buffaloes. So the first question I have, and I just want to make sure, are we able to hear you speak? Dr. Herrera, do we see you? Can we hear you? Okay, so looks like we may have a technical difficulty with Dr. Herrera's line. but here are some questions that have come up. So the first question we have is, what are some specific breeding challenges that you found in Philippines? So you can type your answer in the chat. The question here was, what are some of the specific challenges that you found in Philippines?
Thank you, Dr. Herrera, for your excellent presentations. A minor apology to the audience. Dr. Herrera is joining us from the Philippines, and he's having difficulty with his internet connection, and we are not able to get him on audio. So what we are going to do is to have him respond to us by chat for the questions that come from you, and then we will repeat those questions by audio, either me or Srinivas. Okay? So
yes, Subbu. So I do see the
first question.
Yeah, Subbu, I just
So the first question was, what about future work that you might use the Axiom platform for? So let's give Dr. Rommel a couple of minutes to respond to that. Just a minute. So Dr. Rommel's response to that is he plans to continue using the Axiom platform, and he's planning to use the Buffalo Snip chip to evaluate future selection candidates, namely semen donor bolts. Okay. The next question that has come is, Dr. Herrera, how about what are your plans for using targeted GBS? In the future, this is not directly linked to his use of the Axiom. Let's just give him a couple of minutes, please.
Okay, Subbu, can you hear me? Christina, can you hear? Or Michelle, can you hear? Okay, thank you. So I do see a response from
So his
Dr.
response to that is that he will mainly use targeted GBS if he chooses that platform from us to genotype the heifers. And if we can identify the animals with high breeding value, then he plans to inseminate them using semen from their elite sires. The next question is, what is the direction of his future research with the not just with the Axiom platform, but where is his genotyping work going? Let's give him a couple of minutes.
Okay, we have a new question here from the audience. The question is, how much time have you saved using Snip chip in selecting your approved bull? So I think Dr. Herrera is not able to get our audio. So we will have some of these questions answered via email to all of you. We will send these questions to Dr. Herrera and have the responses sent to you. I think there was a response to regarding the future plan in terms of low density of array design. How do you pick up the markers of interest? And so the answer that Dr. Herrera provided previously was that they are going to look into the indigenous populations and try to use those markers for the design of low density arrays. Okay, I guess given the technical challenges that we are having because of the audio issues, we will get back and please do send us your questions. We will have our team respond and check with Dr. Herrera and get back to you. And we will try to respond to all the questions you have. With that, I would like to announce that tomorrow we have another exciting session. Definitely join us to hear about the applied biosystem AgriSeek targeted GBS applications and how that platform is used for advancing agrigenomics. We do have exciting speakers from the region, and we look forward to seeing you in tomorrow's session. Thank you.